Welcome to LabCast, a podcast about the medical laboratory science profession, brought to you by MedLab Convo. To get notified once we drop an episode, you can do well to subscribe to all our social media platforms at MedLab Convo. For questions, feedbacks, and shoutouts, you can do well to send a mail to at medlabconvolabcast@gmail.com. Thank you for listening.
As a practicing medical laboratory scientist, one statement I hear quite often from patients is, please, I just need the correct test result. To most patients, the laboratory process begins and ends with analyzing their samples. But behind every accurate laboratory result are systems designed to ensure that the result is reliable before it reaches the patient. Two of those systems are quality control and quality assurance. Today, our dear listeners, on LabCast, we are joined by Gideon Oluwatobi Ajala, a biomedical scientist in microbiology department at Royal London Hospital, London, United Kingdom, who will help us dive and understand what these concepts really mean, why they matter, and how they contribute to accurate laboratory results. I am, of course, your host, Jesse Tomlinson, and here with me is my co-host, Pius.
Hi, everyone.
All right, okay, so let's dive into it. Mr. Gideon, quality control and quality assurance are two terms that are often used interchangeably. Could you please explain what they really mean and the key differences between them?
Okay, thank you for having me. Quality control and quality assurance in the medical laboratory is the foundation of every medical laboratory. Clinicians rely on laboratory results to diagnose disease, monitor treatment, and make life-saving decisions. We know the meaning of quality. Quality simply means excellence. You want the best, you want something you can rely on. So those are the two major things the medical laboratory is being worked on, the foundation of every medical laboratory. So the two words are basically used interchangeably, but both means different meaning. Quality control and quality assurance. Quality controls refers to the operational techniques and procedure we use to monitor analytical performance of laboratory tests. Quality control is basically what is being done to ensure that the analytical performance of a laboratory test is accurate, is reliable. So the control is being done along with every of the test performance that is being done in the laboratory. So it answers some questions within the laboratory. Is the test being performed? Is it accurate? Is the equipment, is it performing well? It involves applying the various rules, the Westergaard rules, monitoring the Levigenis chart, ensuring the temperature, the refrigerator, the temperature is being accurate and is being monitored. Now, talking about quality assurance. Quality assurance is a planned and systematic approach that ensures the entire laboratory quality management system is consistent and it produces accurate and reliable results. The quality assurance comprises of the pre-analytical phases, the patient information, the sample collection, the transfer of the sample to the laboratory. It also consists of the internal quality control, the equipment calibration, the standard operating procedure, and also the analytical phase in which the patient samples is being analyzed, and also the post-analytical phase in which involves the issuing of the patient result to the requesting individual, probably the clinicians or just anyone that comes to the laboratory for a test. So the two major differences between the two: quality control involves the procedure that is being done, while for quality assurance, it is a system. It involves both the pre-analytical, analytical, and the post-analytical phase. The quality control, basically, it detects error, while the quality assurance prevents error. Quality control involves is a daily monitoring tool that is being used in the laboratory, while the quality assurance is a continuous quality management system that the whole laboratory system is based on. That's the foundation of the laboratory. Thank you.
Such a calm and elaborate response. I believe most of the misconceptions or the mistakes people make about this quality control, quality assurance is being cleared gradually. And I'm sure by the end of this episode, you will have more understanding as we go on. Okay. Some people may even wonder whether one is more important than the other. In your own opinion, can a laboratory actually function effectively with just one, or do QC and QA work hand in hand?
A laboratory cannot function based on either quality control or quality assurance. No, that is not possible. The two are essential and they are complementary. A laboratory if you say a laboratory has excellent quality control but has a poor quality assurance, that a quality control can only identify, okay, probably the that is being run is not accurate. Whereas for quality assurance, if everything is being done at the analytical phase, probably you have the sample is being done accurately, but in the process of issuing the results, there is a wrong entering of the patient's details or the entering of the results. Definitely, there is a failure with the results. And the same thing with quality assurance. If you have a very good system, but in the process of the quality control, in the analytical phase of it, there is a mistake. Definitely, the whole thing has failed. So both work together. As we have the quality control, we have the quality assurance, which both have to be prioritized and they have to be maintained. Is a standard, is the foundation in which the laboratory is being built on. So without the two foundation, there is no laboratory. Let me just say that there is no laboratory. A laboratory is responsible for producing accurate, reproducible, a precise, something they can rely on results. So if that is not being achieved, definitely quality, we can't say the laboratory is producing a good result, and that laboratory cannot be ascertained to be a good laboratory. So the two works together. Good quality control, but with a poor quality assurance, you can't produce a good result. Good quality assurance without a quality control, you can't have a good result. Thank you.
Wow, wow. And you mentioned the fact that QA, which is quality assurance, is continuous. Do you
Yeah.
think running QC, quality control, and maintaining QA system, quality assurance systems can be time consuming and may involve troubleshooting instruments or reviewing procedures? And then whose responsibility is it to ensure that these quality measures are carried out? Is it the is it only for the quality manager, or every member of the laboratory team is actually responsible for this?
Okay. Uh, Quality control and quality assurance is a misconception by most people to be saying it's time consuming. But if there is a system and everybody works under that same system, then it's no longer time consuming. That is what we are supposed to do. That is why we are a medical laboratory. That is why we are in the lab. It is what we are supposed to do. When you get to the laboratory in the morning, everyone have their own job description, job duties and responsibility. You go to the machine, you run your QC, you ensure this is working correctly. They are part of our daily tasks. So it's not time consuming. Controls, we have the time of the day each has to be done, and a system has already been designed for the quality assurance in which everyone has to work with. Where there is a system, everyone is free. You know what you are supposed to do. You know the time it takes you to do it. You know what are the way forward when the system is not working, or probably you detect an error along the way. You know the action. So where there is a system and everyone works with the system, definitely it is not time consuming, it is not boring, and everyone will work under the system and produce a very good result. Now, to the question of whose responsibility it is for quality control and quality assurance, it is the responsibility of everyone working in the laboratory. Everyone working in the laboratory. It's not the duty of the quality manager, it's not the duty of the director. It is the duty of everyone working in the laboratory assistants, the laboratory technicians, the medical laboratory scientists, everyone involved. It is the duty of everyone. The In some hospitals, the laboratory assistants or the laboratory technicians, they are the one responsible for sample collection. And for every laboratory procedure, it starts from that phase, the pre-analytical phase. If the sample collection is not done correctly, probably a sample is being collected into the wrong bottle. It has already affected the laboratory procedure. It has affected the result already. So if that is not being done properly, definitely the quality system has been compromised. The same thing happens with the role of the BMS who is involved in analyzing of the sample. If the QC or the quality control, the quality assurance system is not being followed, or probably there is an error, that has already compromised the quality of the laboratory. And also the senior laboratory, the directors, everyone involved in the laboratory, everyone has a role to play. When we talk of quality management system, the personnel is one of those. The 12 quality management system, we have the personnel, which are also one of the major factors that makes up the quality management system. Everyone, every laboratory personnel, everyone that works in the laboratory, they have a role to play. Those involved in the training, those involved in the doing the bench work, analyzing the sample, those are the pre-analytical phase, those are the post-analytical phase. I've talked about the sample collection, I've talked about the BMS running the sample. At the post-analytical phase, in the process of issuing the result out, if there is any mistake, probably typographical error, probably swapping of patient results, anything could have happened. And definitely that would cause, um, that would compromise the laboratory system and thereby issuing of a wrong result to the patient or to the clinician that requested for the test. Thank you.
Wow, wow. This is a huge deal in any laboratory setting. A mistake can be a ripple effect for each stage, just like you mentioned, from pre-analytical to the analytical to the post-analytical. Wonderful. So, speaking about frequency, how often should quality control and quality assurance activities be performed in any laboratory setting? Is it something that should be done daily, weekly, or only when problem arise in the lab?
Okay. Thank you. Let's pick it one by one. Like I said the other time, QC, the quality control and the quality assurance, both are the foundation of a medical laboratory. So for quality control, let's pick quality control. Control should be done daily. We have some parts of the laboratory that control has to be done daily. Probably the QC that has to be done on the machine, probably the charting of the temperature for the fridge, the room temperature, and every equipment probably that has to do with temperature in the laboratory. There are some that has to be done monthly. There are some that has to be done probably 3 months, 6 months checks or a year. Let me give you an example of something of such. And also, before I give an example, it all depends on the laboratory and also the number of samples you process in a day. For example, in my own hospital where I work, for urine analyzer, we run over 1,000 urine samples in a day for urine culture and sensitivity. So in that kind of a setting, we can't do control, we can't do it once in a day, the QC for the machine. We do it a minimum of 3 times in a day, in the morning, in the afternoon, and late in the evening. You have to do your QC on the machine. So, and you can't compare a laboratory. Probably there is another laboratory that have the same machine as the laboratory in which I work in. With the same machine, they might decide to do their own quality control daily, probably once in a day, based on the number of samples in which they process. So, but depending on the number of sample you process for quality control, you can design a way, you can design your own method. Probably we want to be doing our quality control once in a day, 2 times in a day on the machine. The charting of temperature, you can make it probably morning and afternoon, depending on the weather or the situation of the environment of where you're working in. The calibration of the machines where you do the servicing. For example, the biosafety cabinet. If a biosafety cabinet, sometimes they do the servicing probably every 6 months or 3 months, depending on the laboratory. So some things has to be done probably daily. Some has to be probably 2 or 3 times in a day. The same thing with quality assurance. We have some daily tasks. We have the weekly tasks. We have the monthly tasks. The daily tasks can include all the temperatures check, equipment check, the documentation review, and everything being involved in the laboratory that has to be done daily. The weekly tasks can include maybe the stock, the equipment maintenance, the review, and like I mentioned the other time about repair of the biosafety cabinet, I said, okay, it has to be serviced probably every 6 months or 3 months, or um, some do their own maybe once in a year. So all those checks has to be done depending on the laboratory. But the most important thing is once a system has been designed, the laboratory has to follow up with the designed method of doing of working, the design method of working. Probably it has been designed that in this laboratory, we do our quality control check probably once in a day for this equipment. Probably the servicing is being done probably 2 times in a year, or once in 3 months. We all have to follow the system. The laboratory must have a standard, must have a way of working. That is why it is called a laboratory. For you to work in a way for you to get an accurate result, a reproducible result, a result that is timely, you must have a system, you must have a procedure, and you must have a way of working. Thank you.
Okay, now let's talk about consequences because you know we've seen the advantages of what quality control and quality assurance is all about. Um, what if this quality control now and the quality assurance isn't properly carried out? How does this affect laboratory results and ultimately patient care? Are there situations where one type of failure has more serious consequences than the other?
Okay. If there is a failure with probably the QC or the QA, definitely a failure of a system will have a serious consequence. If there is a poor QC, it may result into incorrect patient results. Instrument errors may go unnoticed. There could be misdiagnosis, wrong treatment, a delayed treatment, repeating of tests, increasing the healthcare cost. Um, I've worked in a laboratory before in which, while I was in Nigeria, I was working in the hematology department that day, and we've not done the QC we've done the QC in the morning on the machine. The machine was working perfectly. Then there was a break in the machine. We had to do some repair along the line. After the repair, we were supposed to do the QC again. Then we run a sample on the machine. Fortunately, the sample that was being done belongs to a colleague. Immediately the sample was done, the PCV from the hematology autoanalyzer turns to be around 14%. Before we even thought it could be the machine, we were thinking maybe because the colleague was very sick that day. So we thought, this could be the colleague. This could be the result for the colleague. We all went to meet him. Oh, you're not feeling well. We have to, this is a case of emergency. You're having a very low PCV. Then the guy was looking at us like, everything that is happening to me has nothing to do with the PCV. Then we all have to, okay, let's calm down. Let's go back to the machine. Something must have happened. So we went back to the laboratory. We checked the machine. We did the QC. The QC failed. Wow, this definitely has something to do with with the machine. So we had to check the reagents, troubleshoot, and we discovered the error. It was fixed and the sample was done again. From PCV of something around 14, I think it was 14 that day, we got something of, the guy was actually okay. Everything came out perfectly. The PCV was around 40 something. So that's to tell us that QC is very important. Even when there is an error, probably you were trying to work on a machine, there is a breakdown, or you've done a repair. Some machines, once it has been shut down, you have to do the QC again. So the QC is very important. It has to be done intermittently depending on the system that has been designed for the laboratory. The same thing with the quality assurance, which comprises of the whole system. Like I've said the other time, the pre-analytical phase is one of the system involved in quality assurance. If a patient results, a wrong patient details is being written on the sample, definitely, no matter how well you've done your quality assurance, definitely um, that would have led to a wrong patient result. And also, in the process of sample collection, most of the time is one of the issues that happened back in Nigeria. People, most of the time, in the process of sample collection, not everyone involved in sample collection, even the doctors, the nurses, they're not informed about the bottles for each of the tests. I've seen a situation where even the consultants who was asking what is, because most of them, I discovered later on that people who were trained in the 90s, early 2000s, they were not very familiar with the recent bottles, the Vacutania bottles we have now, the EDTL, the lithium heparin, the SST, the various bottles, and they don't really know the importance of taking the sample in the right bottle. So most of the time we see them coming to the laboratory, sending the wrong sample, a sample, the right sample in a wrong container, sample container. So in a situation of such, if that is being taken in the laboratory, you're working towards a wrong result. So in that kind of situation, a poor quality assurance system or a poor quality control system will affect the credibility of the laboratory. Something will definitely go wrong at the long run. So if QC or the QA should fail, definitely there is a bridge in the patient results and the reliability of the results. And there is something I discovered along the line. I came across the statistics that says 60 to 70% of lab errors occur during the pre-analytical phase. And based on my laboratory experience, I can assure you that this is very true. Samples are not collected properly. Most of the samples being sent to the laboratory are not being taken properly. But if there is a quality assurance system that ensures the right person that has the right information about how samples should be taken and does this, definitely we will be able to have an edge in tackling this error. Thank you.
Awesome stuff. Awesome stuff. Wow. You have a lot of experience and everything. So I'm going to piggyback on that experience now. From your years of practice, have you encountered situations whereby poor quality practice affected laboratory testing? And if so, without breaching patient's confidentiality, could you share an example and what lessons were learned in the process?
Okay, thank you. There were a lot of situations that has happened along the years. There is one I remember vividly. I was explaining something the other time about a consultant asking questions about the various bottles being used in the laboratory. While I was working in a teaching hospital in Nigeria, there was a time a sample was sent to the laboratory. So when they send the sample to the laboratory, we check the bottle. Those are the reception. They check the sample bottle. They check the patient details. They check the test on the form of the sample. Is the doctor requesting for the right test? And has the sample be collected in the right bottle? So in that kind of situation, when we discover that, oh, the sample has been collected in the wrong bottle, we just give the patient back the bottle and tell them, oh, this sample, we can't collect this sample from you. Probably the sample is being taken in the wrong bottle. So The patient will go back to the ward or to the doctor to tell them about it, that, okay, they rejected the sample from the laboratory. They told me that this sample has been collected in the wrong bottle. And you know what they do? Okay, they said, oh, they should take the sample in a probably EDTA bottle or lithium heparin bottle. They will pour the sample from this previous bottle to the new bottle and then give it back to the patient and tell the patient to bring it to the laboratory. So when we discover that, if that happened to a patient, we gave the sample back to the patient. Okay, take it back to the doctor. This sample was collected in the wrong bottle. When the patient came back, we noticed. We now asked the patient, did the person, the doctor or the nurse that took the sample from you, did they pour it back to the bottle? The patient said yes. Oh, this is what they do when we tell them to take the sample back. Okay, now we have a system now. If you are sending a sample, if you send a sample to us in a wrong bottle, we will not return the sample back to the person or probably to anyone that brings the sample to the laboratory. We tell them, we write a note for them, take a new sample in this type of bottle and send it back to them. So that actually worked, and they were able to, we were able to achieve a good thing, and also reorientation, explaining to them, okay, if you want to take a sample for this process for glucose, you have to take the sample in a fluoride oxalate bottle. For lithium, for other chemistry tests, you have to take it in this type of bottle, lithium heparin bottle. The same thing for microbiology test. Everything has to be done the right way. So I've had an experience of something of such before, in which a wrong sample was taken in a wrong bottle. And I can tell you, this is not a problem in Nigeria only. It happens almost everywhere in the world. Most of the time in the laboratory, while in the UK, we receive sample in a wrong bottle. We receive sample, probably they're requesting for a wrong sample being sent to the laboratory. They might be requesting for probably a urine sample, a urine culture, and they send you a stool sample. They might take a microbiology sample, and then the sample is being taken in a virology bottle. So it happens everywhere. And one of the best way of correcting this thing is to sensitize the people involved, where the sample is coming from. Let them know. And another thing we did was to give them the phone number of the laboratory. Anytime you want to take a sample from the patient and probably the person, you don't know the right bottle or what to do, you can give a call to the laboratory, ask questions, and we will direct you on what to do. I want to take a CSF sample. What type of sample, what type of bottle should I use? I want to take a sample for this test. What type of bottle should I use? Can I make use of this? And often time of the time, they make a call to the laboratory to make this call. I am making call to the laboratory to ask these questions. Thank you.
Wow, wow. When you cited your example, I was laughing over here because um, they said no system is completely perfect. I think in the lab where I do my posting, my clinical posting, we do accept we reject the sample and ask them to go with the container, not knowing what happens later on. But you know, that itself tells us that no system is completely perfect. What may have been done in your case may not be done over here as well. So what happens when quality control itself fails, or when problems in the quality assurance system goes unnoticed? Are there safeguards that help detect these issues before incorrect results are released?
Yeah. Thank you. No quality system is perfect. And that is why there is a there is we have a multiple safeguard to minimize the risk. Although we have a system, and I can assure you there is no system that is perfect. Even in the UK where I work now, we do a review almost every week. There is a review of the system. There is a review of the workflow. There is a review of the methodology. And the reason is because we want to try as much as possible to be able to meet up with the standard, to be able to correct some of the things we're doing. And that is why we have the internal quality control, we have the external quality assessment, the EQA, and the high QA. The EQA is to test our process. Most of the time, when a sample has been processed before, probably a sample with we have some samples that we get some values that are very high. We have some that we get some values that are very low. We have some organisms that are being isolated that are rare. So we keep some samples, we save them, and then we use some of those samples as internal quality control for ourselves. So having those kind of safeguards, it helps to minimize the risk of issuing or probably to minimize the risk of a of a um, of a failure to quality control. So we have the IQC. This is done regularly, probably weekly. It could be weekly, it could be bi-weekly, it could be monthly. So just once in a while, the quality control manager, we get some of this sample, we send it in. It will go through the right channel. It will be sent to the laboratory same way we receive other samples, and then it will be processed alongside every other samples that are being received in the laboratory. And then the quality manager she already knows the sample, she has the results, she has everything. So she will be able to know, okay, we were able to get the results from this sample 3 weeks or 4 weeks ago. After 4 weeks, it went through the same process and we were able to get this value. So they will do a comparison between the two results and they will be able to say, okay, definitely we're making a progress. Definitely our QC is working perfectly. So I can assure you there is no perfect system, but there are safeguards. There are methods of doing it, and an example of it is the IQA that I've just explained. Another thing is the external quality assessment. We have some external quality bodies. In the UK, we have the NEQUAS. In Nigeria, I know the MLSCN, they are responsible for accreditation of laboratories in Nigeria. They send samples to different laboratories in the country. They send it to them. They ask them to process it, forward the result to them. They already have the result. So each laboratory will process the sample and send the results back to the regulatory body or the body that is being involved. And this has to be done regularly, and no laboratory should shy away from receiving that kind of a sample. And then I can assure you, even in the UK, there are situations whereby we fail external quality assessments. They will send a sample to us and we will miss one or two things from the sample. But that is to tell us that, okay, what is happening? What has led to the failure of the laboratory results? And there are ways to go about it. We review our procedure, we review our methodology, and we try to see, okay, where are we missing it? What can we do right? So those are the ways we can actually work on quality control failure or quality assurance failure. Another thing is repeating of tests. I can explain the IQC. We can make use of an IQC, probably a test that has been done before, or there could be an internal audit. We do that most of the time where I'm working now. We rotate people on different benches. We have a senior who go to the various benches. They get to see what people are doing, they get to see the procedure, they get to see how it is working, they get to test the reagents, find everything that is happening, what are the risks, what are the things that can actually cause a result not being accurate based on the procedure we have. And also we have the corrective and preventive actions that are being done. We don't wait for a problem to occur before you solve it. The same thing when we're doing a risk assessment. You don't wait for a risk or an accident to happen before you try. Anytime you notice this could definitely lead to this kind of a problem, you fix it at that point. You don't wait for it to happen before you fix it. You have to be proactive in a way of solving problem. You don't wait for the problem to occur before you now think of what to do. You should be proactive. You should be able to project based on the procedure and everything being done in the laboratory. Thank you.
Awesome input. Thank you so much for the elaborate response. So in Nigeria, Nigeria that we know of is, of course, a low medium income country, and many laboratories, particularly in the rural settings, work with limited resources. So they rely mostly on rapid diagnostic tests, the RDTs, which can occasionally produce false results. What quality measures can laboratory put in place to improve the reliability of these test results despite these challenges in the rural community?
Okay. One of the things, based on my own observation, my own experience from Nigeria transitioning to the UK, what I've experienced is that we we tell a lie to ourselves. We don't have the equipment, we don't have the apparatus, we don't have the finance. But I can tell you, you don't need a lot of money, you don't need a very big place before you produce a very good and reliable, accurate and reliable result in a laboratory. We know Nigeria is a low income country. We know we rely on some of um, the simple methods of laboratory. I would say the conventional way of doing laboratory procedure and testing. But we can still maintain quality. We can still work very well. Let us pick an example of the rapid diagnostic test kits. Rapid diagnostic test kits, they are different. Let us pick an hepatitis B, for example. We have different manufacturer of hepatitis B test kits being produced in Nigeria on the market. We can try as much as possible to get, okay, which one is the best? Which is the best product? Which product is more reliable? We are in the region in which our temperature is definitely between 30 to 40 degrees. We have a laboratory that we don't have electricity for 24 hours. Which test kit will be able to give us the kind of results we want and be able to work properly under this kind of temperature? After that, then we design a standard operating procedure. Those are some of the things I observe we don't do properly in Nigeria. Our SOP is just, okay, add 1 microliter of this to this. SOP is not like that. SOP should be like a textbook. It explains every details, everything you need. There should be standard operating procedure. Even if you want to discard, there should be standard operating procedure for discarding waste in the laboratory. We might see it as, oh, you just discard your waste here. No, there should be ways of doing it. There should be a way. So when a kit is being purchased, we must we must have our own standard operating procedures. How do we want to use these kits? How do we want to get a accurate result? We must design a quality system. We must design a quality control for the methodology for the hepatitis B test kits we want to do. So, having purchased the kit, I'm using this as an example. I want to be doing hepatitis B test in my laboratory in Nigeria. The first thing to do is to observe, okay, which kits will be able to work well based on the temperature, based on the laboratory that I have. I don't have 24 hours electricity supply, but electricity supply is within 10 hours. Okay, I'll be able to store my kit in this place. When I bring the kit out in the morning, I have to keep it on the bench. What's the room temperature? You must know every details of the environment you're working in. And then once the test is being done, some of the sample that are being tested positive, you can send it to another laboratory to do a ELISA technique on it to be able to tell you, okay, definitely your kit is working properly, and your kit has a range of this between these days to this, probably after the patient has been exposed to hepatitis B between these days to these days, your kit can pick it. Once you have everything, you know everything about the kit you are using, then you can set up a laboratory, you can work within that means. But the problem we have is that we will just believe we don't have the resources, we don't have this, we don't have that, so we can do things as we like. We think we are helping the patient, But I don't think that is right. We should prioritize quality, accuracy, reliability ahead of just meeting the patient's need. If you're able to issue out a result and you are not giving out the good result, a quality and an accurate result, definitely is more or less like not giving a result. So one of the things I think we can do is we should prioritize the way our methodology, our procedure, we should prioritize quality. We should prioritize the way we work, the way we do our procedures, our purchase, inventory, every system that is being involved, and the personnel that is being involved in this procedure. We must make sure everyone is well trained in this procedure. We must maintain a proper documentation. That's another thing I noticed in Nigeria. Most laboratory, although I know most laboratories do not have a laboratory information system, but at the same time, even without a laboratory information system, you should have a system in which you can do a documentation for every sample that comes to the laboratory. You can say, okay, will we be able to keep this sample and this result a documentation? We can do it for 2 years, 3 years. It's not expected of you to be able to keep a result forever. Even laboratory information system, the ones being used in the Western world, they don't keep results forever. There are some that can tell you, okay, we can keep the patient result and the patient details for 10 years, 15 years. They can't keep it forever. So just have a time frame. We can work within our means if we are ready to achieve quality. Thank you.
Hmm. It seems like the question I'm about to ask now is more or less like a build up and a need for more emphasis on what you said. So let's talk about staff training and tech. First of all, on staff training, what do you think? How important is continuous staff training in maintaining quality, strong quality systems? And then, looking ahead, do you think automation, machine learning, or artificial intelligence will play a role in strengthening quality control and quality assurance in medical laboratories?
Yeah, thank you. Continuous staff training is very important and is essential in a laboratory. Even if you've been trained on a bench, there should be a time frame for a review. You've been trained on how to do this procedure. There should be a time frame. Okay, after 2 or 3 years, you should be retrained on the bench. While I was in Nigeria, I think we don't really prioritize continuous training, which is one of the things that we overlook. Someone has been trained on this bench and you expect the person to just do it 5, 6 years, they're still doing the same procedure. It's not like that. Where I work now in the UK, we do a review of training every 2 years. You can be trained on a particular procedure for 3 months, 6 months. After that has been done, you are expected to feel a competency. It's just like a questionnaire. You answer questions. It has to be signed by someone, your trainer. And once that has been done, after 2 years, you are expected to undergo the same training again. So that's to tell you that the fact that you've been trained, training does not have to stop. It has to be continuous. You have to be retrained. Probably even if you're working on the same bench for 2, 3 years, there should be training. And also, external training is also important. It's important that people go for uh, conferences, trainings outside of where they're working, just to get themselves exposed to the new procedure, the new methodology, how things are being done now. What are the things you need to know? What are the new invention in laboratory practices? So it's always important for everyone to stay updated. Everything is changing now. New machines are coming in. You have to get yourself involved. You have to know. You must know what is going on. You must know the latest trend we have in testing and analyzing of a particular procedure. And also talking about automation and AI. definitely automation and AI has an improved uh, as a way of improving quality control in the laboratory. For automation now, most of the laboratory procedures, everything is now based on automation. And I can assure you, one of the things automation has brought to the laboratory, it has reduced manual errors. I was talking about laboratory information system the other time. Anytime a sample is being done, as you're working on the sample, you have everything is going to the laboratory information system. And from the laboratory information system, the result is being sent out to the doctors directly. And most of these machines now, they are connected to the laboratory information system, to the LIMS. And from the laboratory information system, the result is going directly to the doctor. So therefore, manual errors are being eradicated from that kind of a procedure. For example, the chemistry analyzer, most of the ones they use in the UK now, and even in the ones in the UK, most of them are connected to the laboratory information system. So once you have your sample, load your sample to the machine runs the sample and send the result directly to the clinician involved or the person that requested for the test. It doesn't go through any other channel. So the same thing with urine automation that is being done now. Once the urine microscopy is being done, if the sample needs a culture, the machine will tell you proceed to do culture. If the urine sample does not need a culture, it gets sent immediately to the person that requested for the test. So based on that, definitely manual errors has been reduced. And also automation help to improve consistency. When we talk about laboratory, we talk of accuracy, we talk of precision. Precision means in the sense that you have a sample now, you're getting your results, and your results are close to the true value. Like that's accuracy. Like we have a closeness between values. If a sample is being done now by a machine and you get 34, if it's being repeated several times, there is possibility that you will keep getting the same value. But probably if the procedure is being done by human, there might be variation in the sample results. So that has definitely improved. And also traceability. Probably there is a bridge, something has gone wrong along the way. With the use of the automation now, you can just go into the compute to the machine, search the sample, what's wrong. It will tell you what has happened, why the sample is still hanging, the result is still hanging, why it has not been sent, why everything is being worked on. On the laboratory information system now, we have a way to check. For example, you've received a sample in your laboratory, 2, 3, 4 days, the sample has not been processed. While I was in Nigeria without laboratory information system, you may not know that a sample has not been worked on 2, 3, 4, 5 days, may probably until when the doctors comes to the laboratory to tell you that we sent a sample some days back. Yeah, when did you send it? Who did you give it to? Most of those things no longer happen with the use of laboratory information system. And the reason is because any sample that is being booked in, that is being received in the laboratory, you have the details of everything on the computer. So you can go through your incompletes. Just go to the incomplete on the laboratory information system. It shows all the samples that are yet to be released out. And then you can check, why is this one not yet released? What's delaying the result? And you can work on it and send the result to the person that requested for the test. So the same thing with artificial intelligence. Artificial intelligence now has helped laboratory procedures a lot. And the reason is because of the the accuracy. It can access information, a lot of information. They make use of artificial intelligence now in histopathology. Most of the hospitals, laboratories we have now in the Western world, they make use of a digital pathology where slides are being scanned. Once the slide has been scanned, then they have the artificial intelligence that helps to run the scanned slide and then check it with a database and tell you, oh, this is this is the diagnosis of the patient. The patient is having cancer. The patient is not having cancer. Even most of the machines now, they're incorporating artificial intelligence into it, and is drastically improving laboratory results, drastically improving diagnosis, and is reducing some errors, manual errors, human errors that occurs in the laboratory, detecting of errors too. Probably when a quality fails or something is wrong with the machine, most of these automation, artificial intelligence, they help to detect this and they tell you what is wrong, the next step of action to take. And that's where the world is going. And I'm happy about it as a practicing scientist. The reason is because the importance of a laboratory is to be able to produce the accurate, reproducible, and reliable results. And when accuracy is being achieved, I can assure you it helps the patient. Early detection offers the best part of a cure. And laboratory is one of the major, we account for 70% of diagnosis being made in health sector. So it tells you that when a result is being accurate, when we are getting it right, we are helping the patient, we are doing the right thing, and we are fighting the infection, we're fighting whatever is happening, and we are solving problem. Thank you.
Thank you so much. Yeah, every part every part of the QA and QC is actually very important and should not be neglected. So I want to put you on the spot right now. If there is one misconception healthcare professionals or even young medical laboratory science professionals have about quality control and quality assurance, what would you like to correct about this misconception?
Okay. Yeah. Like I explained the other time, one of the major misconception, I had a misconception, and that's the reason why I can tell you about it now, is the fact that we believe is a robust system. We believe we need the money. We believe we need the equipment. We believe we need the machines to be able to achieve this thing. But I can tell you, you don't need a big environment. You don't need a big machine. You don't need elaborate equipment. Even the conventional way of running a laboratory, you can still maintain quality. You can still work with you can still embed your quality control and quality assurance system. You don't need a big laboratory. You don't need the latest machine. You don't need the latest equipment. Just do the basic thing. Do it correctly. Review your procedure. Review your methodology. Review the way you do things. Try to observe what can we do to improve the way we work. What can we do to improve the way we diagnose, the way we run our procedure in the laboratory. And with that, I can assure you, you will definitely be able to achieve most of these things. It's not a big system, it's not a robust system as we think it is. It's not something that is not achievable. It's not something we cannot do. We just have to be intentional about it. We have to make sure we want to do the right thing. Our laboratory must be able to produce the right results. The small machine you have, the conventional way you were doing it, do it the right way. Have the right SOP on your bench. Be ready to improve on yourself. See the way things are being done now. What can we do to improve our methodology? What can we do to improve our accuracy? What can we do to improve the time in which the result is being released out? We have to be intentional about it. And I can assure you, if we are intentional about it, we take the right step, we we do the right thing, we work as a team, we enlighten ourselves, we are ready, we are open-minded, we are inquisitive, we can achieve it and we can do it. Thank you.
Yes, that's the spirit. That's the spirit. Intentionality, teamwork, everything comes together. They align together for this thing to be successful. Oh, okay. So, and that's it for this episode, dear listeners. Oh, thank you so much, Mr. Gideon, for joining us and for sharing your experience with us today. Today's discussion reminds us that producing accurate laboratory results is much more than analyzing a sample, Behind every reliable result is a system of checks, standard, and dedicated professionals working to ensure that patients receive results that they can actually trust.
Thank you for tuning in to LabCast. If this conversation resonated with you, I, I mean, it did with me, so it should to you too. Please share it with your lab network and join the discussion using hashtag LabCast. You can email us your thoughts at medlabconvolabcast@gmail.com and follow us on all platforms at medlabconvo. Until next time, protect your peace and stay curious. Bye. Bye.
Bye.